The retroviruses are a fantastic tool to do gene therapy. So gene therapy is a way to treat genetic disease. So this is a case where somebody has a mutation in a gene and that leads to a protein in their body functioning poorly. And if that's an important protein, that can lead to illness. And so you can treat that in various ways, but a really good way to treat that disease is to fix the gene defect in the cells that need that protein. Another use of gene therapy, which is becoming increasingly effective, is cancer. So cancer cells are doing different stuff to your normal body. The cells in your body, they're dividing very rapidly, they're sucking up a lot of metabolites, their tumor might be growing blood vessels and stuff like that. So you can put in gene therapy to change the immunology of the tumor so that your immune system can see the tumor again. Where the tumor has kind of shut down its ability to be seen by the immune system, you can fix that with gene therapy. So gene therapy essentially involves the ability to infect your target cells with a virus, and that virus must contain the gene that you want to put in there to either fix them or to make the tumor visible to your immune system, for example. So you can use retroviruses for gene therapy because you can firstly make viral particles with the genome inside that only contain your gene, your favorite gene. And you can then infect your target cells, and those infected cells will only be modified by the insertion of your target gene into their chromatin. So that's great, and retroviruses are particularly good at that because they insert it into the chromatin and so it's stable. Once the gene's there, it's stable. So with all gene therapy, the rate limiting step is simply the ability to put the gene into the right cells. So the gene therapies that work best are often the ones where you can either inject the virus directly into the site, so for example into a tumor, or the case where you can take the cells out of somebody's body, so for example their blood cells or their bone marrow cells, modify them by infecting them with the viral vector, and then put them back in their body. And in that way, there's a relatively straightforward way to modify people's cells with a gene therapy vector that's derived from a retrovirus. So in the early days, gene therapy was carried out with the mouse gamma retroviruses. So that was taking a mouse virus, taking out the mouse virus genes, putting in your gene, and then using that to deliver the gene. And that's worked very well, and there's still gene therapy trials going on using the mouse virus. The problem with the mouse retrovirus is it will only infect cells that are actively dividing, and there are many cells that you want to infect that aren't particularly dividing, so stem cells don't divide very well, and there are a variety of other blood cells that don't really get infected with the gamma retroviruses. So more recent therapies are using HIV-1 derived viruses. So you can imagine that that can cause a problem because you don't want to infect somebody with a virus that's going to cause them any additional disease. So there are a lot of safety concerns with retroviral vectors, and people have been very careful to disable the virus in multiple ways so that you can make it safe. Of course, disabling the virus makes it less infectious, so there's a constant trade-off between how safe the virus is and how effective it is as a gene therapy vector to deliver a therapeutic gene. So the history of gene therapy has really gone from developing the vectors to be more sophisticated, safer, and more infectious. And then the other problem is engraftment. So you have to be able to put the modified cells back in the person's body and not have those cells immediately die. So testing various ways to do the infection, how you look after the cells very carefully after you've taken them out of the person's body, how long you keep them out of the body, what media you grow them in and stuff like that is a massive industry with people trying to work out the best way to take the cells out and put them back in without damaging them in any way. So one of the most exciting gene therapies that are going on at the moment are what are called CAR, or chimeric antigen receptor. So that's based on the idea that when you get a cancer, so I think everyone agrees now that cancer is something that is really fought by your immune system. So during your life, cancers are forming on an individual cell basis, but they're killed. So cells do strange things, and your immune system recognizes something funny is going on and kills them. So in order for a tumor to survive in your body and grow, it has to change in such a way that your immune system can't see it. So that is bad news, and if your immune system can't see it, the tumor can grow. So the idea of CAR therapy is that you can take the cells out of somebody's body. They're T cells, which are the cells that might be killing tumour cells, and you can modify their specificity so that they can see the tumour again. And they do that by making what is referred to as chimeric antigen receptor. So essentially, the T cell receptor, which is on the top surface of the cells, swimming around looking for things to kill, you change that molecule for an antibody that specifically recognizes the cancer. And you fuse that antibody to the other half of the T cell receptor, the part that's inside the cell, so that when the antibody on the surface of the T cell binds the CAR, the chimeric antigen receptor, then it activates the T cell to kill the tumor. And that's a fantastically effective therapy when it works. And there are people who've been treated in 2015, 2016, children with leukemias that would not be treatable in any other way, and yet the CAR therapy can give them effectively a cure. So that's a really exciting area of gene therapy that's being developed at the moment. It's really starting to work. So because gene therapy is a way to cure a genetic lesion, sometimes it's the only way to treat a particular disease. There might not be any other strategy to do that. So one of the real values of gene therapy, I think, is it brings a whole new series of treatment options. Unfortunately, as it develops, it's very expensive and it's very bespoke. So you often find that you can only treat a small number of patients per year, and you might have to have that person in the hospital, you might have to take cells out of their body, you might have to make a treatment that is really specific to their disease. And so at the moment, gene therapy is not something that can be mainstream, that can be used to treat thousands of patients in the way that surgery or other treatments can. So I think that's one of its great disadvantages. But as we develop gene therapy, then there is every chance that it can become mainstream therapy. And I think gene therapy really is the future for many things, not just genetic diseases in the future. So for example, there are ways to try and treat HIV infection with gene therapy. And the idea would be that you might you might take cells from someone's immune system out of their body and then make them insensitive to HIV infection by, for example, stopping them making the HIV receptor CCR5, or by expressing an antiviral gene that is part of your innate immune system. So a modified innate immune factor, put that inside the person's T cells, put those T cells back in their body, and then those T cells, those modified cells, won't be infected by the virus in their body, and so those cells can fight the viral infected cells. And it's possible, theoretically, that could be a one treatment cure. But we're a long way from that, and it's about improving our ability to deliver the genes. It's about selecting the right genes to treat each particular disorder, and it's about getting better at the actual delivery of these genes to improve the efficiency of delivery and the efficiency of engraftment, so the maximum number of cells are returned to the patient effectively. So there are some risks with gene therapy, and there are cases where people who've been infected with gamma retroviruses encoding a therapeutic gene have ended up getting cancer. And it's not entirely clear why that is. It's a combination, I think, between the fact that the therapeutic gene had certain properties and the gamma retrovirus had certain properties, and when those properties are put together, that significantly increases your risk of getting cancer. So those studies that discovered cancer in children that had been treated for immune deficiency syndromes, those studies were very worrying, because people thought maybe this will kill gene therapy, maybe we'll be in a situation now where the risk for cancer will be too great. So it was very important to do gene therapy in a different way, and in a way that will not cause cancer. And I think that is achieved by using a slightly different therapeutic gene and by using a different vector. And the shift to using HIV-based vectors is driven in part by the fact that they are less likely to cause cancer, because HIV typically does not cause cancer. So it's very important to understand with gene therapy that we're not infecting with a virus that can replicate. So each viral particle will go into a cell, either in your body or in the lab, depending on how the gene therapy is done, and it will deliver its gene to the nucleus of the cell, and then the viral particle is gone, and there is no opportunity to produce new viral particles because the genome of the virus has had all of the viral genes taken away. So it's what we call a single round vector. So you can infect cells and they'll express the gene you put in there, but they won't make any virus and they can't infect any other cells. So one risk in the early days was that if a person who's been treated with gene therapy using an HIV vector, what if they get infected with HIV through the normal sexual transmission? So that may lead to a situation where the virus that's in their body contains a mixture of genomes, the genomes of the virus that infected them plus the genome of the gene therapy genome. Then they could infect another person and that modified genome could transmit to them. So we've worked out a way to stop that happening, so that there are bits of the virus that you can cut out and the virus still works, but a new virus showing up cannot package that into viral particles. They're called self-inactivating mutants, and they're very safe. So one of the problems of doing gene therapy with retroviruses, particularly with HIV, is that we have to transfect cells every time we do it to make the vector. So that means we have to get a cell line in the lab and we have to put it into flasks and we have to introduce the viral DNA into the flasks fresh every time. And it makes it very expensive, and each time you do it you have to check that that particular prep is safe, and you have to do lots of regulatory tests on it, which uses up some of the prep, and you have to remake it every time you want to treat a patient. What we would like to do is to develop a cell line that contains the gene therapy vector and constantly produces it, and then the virus that's coming out of those cells could be used as a medicine, and you can grow those cells for a period of time and keep making fresh virus. That requires to develop a cell line that consistently produces large amounts of retroviral vector. And that is one of the holy grails of the gene therapy field, to develop these kind of reagents that will constantly provide a consistent and a safe source of the retroviral vector to put into people's bodies. So I think that's one of the great challenges. Another great challenge is developing each gene therapy case. Each disease will require a new gene. So you have to figure out which gene you want to use, what strategy you want to use in order to treat the patient. You need to be able to infect the right cells. And so there's a great deal of work goes into developing new ways to use established gene therapy techniques to treat new diseases. I think, as I said earlier, one of the most exciting features of that work is the use of gene therapy vectors to treat cancer.